Hello, everybody, and welcome back. So it's a huge pleasure and privilege right now for me to introduce our first speaker in this series. And while I do that, Dr. Jha, please feel free to unmute yourself and start sharing your slides as well. So Dr. Ajish Jha is an assistant professor of biology at New York University in Abu Dhabi, where he leads the Genetic Heritage Group. Dr. Ajish grew up in Kathmandu, Nepal. He received his bachelor's in molecular and cellular biology from the University of California, Berkeley in 2007, his PhD in human genetics from the University of Chicago in 2015, and completed his postdoctoral research at Stanford University in 2019. Currently, his research focuses on integrating population genetics and microbiomes of traditional populations in the United Arab Emirates, Nepal, India, and Oceania. His work has spanned the microbiome, genomics, bioinformatics, immunology, evolutionary biology, and cancer biology, which has resulted in multiple peer-reviewed papers. He has received funding from NUAD, Stanford University, as well as the Bill and Melinda Gates Foundation. So thanks so much for presenting today. And yeah, please take it away, Dr. Jha. Dr. Jha, I think you're still muted.
Sorry about that. Let me share my screen. Can you see my screen now?
Yes, looks good.
Okay, thank you so much for a great introduction, and thank you for the opportunity to come and talk today. It's super late here. It's actually midnight, so I may be a little bit rusty in my Zoom skills. But today I'm going to talk about scaling up multi-omics studies to identify functional contributions of microbiomes on health of underrepresented populations. And those of you who are here in the audience, you probably know that this is super important. That's probably why you're here. So the last few decades of research in human microbiome has been very productive. And what we now know is that gut microbiome varies significantly across lifestyle, right? So in this paper published in 2017, this group of researchers, they looked at gut microbiome variations across a lifestyle gradient. Each dot here is an individual. They're color-coded by the populations they come from. Lighter colors are populations or that are practicing traditional lifestyle. Darker colors are populations from industrialized nations. And as we can see, there's a stark differences in gut microbiota across these populations. Recently, we published a paper in which we looked at gut microbiome variation in children. And what we found was that initially the gut microbiota of children across different lifestyle doesn't vary much. But by 3 years of life, the gut microbiome is starkly different. So in this right-hand side plot, the circles, the red circles indicate individuals coming from non-industrialized populations like the Hadza. The blue, light blue color dots indicate populations coming from transitional populations, and the blue, dark blue dots on the top, they represent children from industrialized populations, right? So there is a massive variation in gut microbiome across lifestyles. Oops. What is going on? Okay. But what we also know is gut microbiome development is an ecological process. So in this particular study conducted in Italians, what the researchers found was initially, if you look at the circle in the middle, the pie, initially in a child, a one-day-old child's gut is colonized by different types of bacteria that come from a variety of sources. The red outer circle indicates bacteria in the child's gut that look like an adult gut microbiota. By 4 months' time, though, more than half or about half of the child's gut microbiota resembles the microbes in an adult gut, right? So what this means is initially microbes are like, hey, look, it's an open space, let's go make home. And they are stochastically, they're just, every microbe is trying to colonize the gut. But many of the microbes are not very successful in this colonization process. So by 4 months of time, the successful microbes are the ones that are traditionally known to live in the gut, right? So this process is very important. By 3 years of time, the child's gut microbiota starts to resemble an adult microbe. What we know is that the adult microbes interact with many human physiological processes. So in this figure on the top in the green section is gut lumen. That's where all the microbes live. In the middle, the cells with finger-like structures, these are epithelial cells in the gut. Those cells prevent microbes from getting into our bloodstream. Although the microbes cannot get into our bloodstream, they produce a variety of different chemicals that can get into our bloodstream, and these chemicals can go and influence a variety of different physiological processes in our brain, in our organs, and whatnot. So the imbalance in any gut ecosystem can lead to disease. So there's a paper that came out from Sean's group, actually, the Monod et al. Paper. They looked at association between gut microbiota, individual taxa, and a variety of different human phenotypic traits. Some of them related to disease on the top, shown by orange box. Some of them are related to a healthy lifestyle on the bottom. So red means, so in each column is a bacteria, each row is a trait. Red means the bacterial abundance is elevated in these traits or is positively associated with those traits. Blue means they are negatively associated. So we see a whole bunch of bacteria that are associated with disease and healthy traits, and they are sort of opposite to one another, right? So when they are positively associated with health, they are negatively associated with disease-like phenotypes. This indicates that gut microbiota are strongly associated with markers of human diseases. Gut microbiomes are also associated with treatment efficacy. In the right-hand side plot, Zimmerman et al., they looked at 76 gut bacteria isolated from the human gut, and they asked, can these microbes metabolize drugs? So after 12 hours, they found, so black means 0% of the drug was metabolized, white means 100% of the drug was metabolized. If you look at the top and the bottom of this heat map, you can see that there are a lot of white regions, right? So some drugs are metabolized by all of the bacteria on the bottom. On the top, some of the drugs are metabolized by some of the bacteria, and a lot of the drugs are not metabolized at all. So what this means is, given your microbiome composition, gut microbiome composition, the treatment efficacy in a diseased individual may vary. So this type of research has led scientists to sort of come up with sort of like a personalized nutrition. So in this particular study, ZV et al., they looked at or they measured a variety of different features in 800 participants. One of the features was microbiome, and by putting all these features together, they were actually able to design personalized diet to lower glycemic responses. There are actually startup companies out there today that use either your genetic data or your microbiome data to design personalized nutrition. And these companies are expected to have a massive impact in healthcare. So this is great, but all of these, ecological succession, association with disease, personalized diet, is only relevant currently to people of European descent. Why is that? Because most on the top plot here, in this paper published earlier this year, what this group of scientists found was that most of the samples on which microbiome has been characterized come from European individuals or individuals of European descent, right? Even though Europeans constitute a very small fraction of the entire world population, most of the samples come from Europeans. This is improving at the bottom. They looked at what proportion of samples come from different world population over time. 2010 and 11, 100% of samples came from Europeans. In 2020, there is a little bit of diversity there, but still dominated by Europeans. But I think the problem goes beyond the number of sample or sample proportion. I think there's a problem in how we sort of approach microbiome studies. So the first step in microbiome study is characterizing the microbial variation. Second step is to then say, okay, if there's some variation exists, is this variation associated with some trait, like the Monod et al. Paper? The third step is, okay, there are a bunch of microbes that are different. We could predict their function, but to make sure that these predictions are true, we need to layer additional omics data sets. So these omics data set may be metabolomics, metatranscriptomics, metaproteomics, whatnot, right? So then we understand by layering all these different omics data sets, we then understand what microbes are there or not there and what they're doing or not doing. And then that sort of leads to this translational research that can help the human society at large. In the case of European microbiome studies, all these are happening and translational studies are being done at a very rapid pace. But in traditional microbiome studies, we stop at characterization. The number of samples are increasing, but they stop at characterization. There's not a lot of studies looking at association between microbiome and traits, microbiome and disease, not a lot of studies looking at functional data, and very few translational studies. So then the question is, why is that? One answer could be the prevalence of chronic diseases are very low in traditional populations. Second scenario could be that chronic diseases exist, but they are not associated with microbiomes in traditional populations. The third option or third scenario is that both of these are wrong, but there are so many barriers to conducting this sort of functional or translational studies in traditional populations that we never get there. So last few years of my research has been focused on trying to figure out which of these scenarios are true. So what we have done is we've visited a number of traditional populations in Nepal, which is the Himalayan country. So on the top left is a photo of one of these traditional villages. We've sat down with the community leaders. We've discussed with them the scope of our study, the scope of microbiome study, the importance of it for participating populations. We've collected a lot of survey data, sort of characterizing people's diet, use of medicine, and whatnot. And we've also measured some phenotypes. And the analysis of this data shows that chronic diseases are prevalent in traditional populations. So in this plot, the histogram plot on the left, what we found was systolic and diastolic blood pressure both are highly prevalent in the indigenous populations of Nepal, who are severely underrepresented in microbiome, genomics, both literature. BMI, or sort of like high or overweightedness or obesity is not that high, but hypertension is very high. And if we look at neighboring country, India, what we find is that hypertension is massively prevalent. In fact, cardiovascular disease is the #1 non-communicable disease that causes deaths in India. High blood pressure or hypertension is the primary risk factor for CVD, and hypertension is highly prevalent among the poor and marginalized populations. So on the right-hand side here in the map is a sort of representation of prevalence of hypertension in India. Red means 10 to 15% hypertension, right? So almost all of India is red. On the left-hand side or right-hand side within this green circle is a state of Assam, where hypertension is prevalent at 15 to 25%. So we collected samples in Assam, where our collaborators in India did this study. They collected the samples, they generated 16S data, and I helped them analyze this data. And in a paper that was published earlier this year, we found a robust association between systolic blood pressure and microbiome composition, indicated by PICO1 on this plot on the left. right? So what this means is chronic diseases are prevalent, and at least hypertension is associated with microbiome, even in traditional population of South Asia. So the third scenario seems to be the true scenario, where there are many barriers to conducting microbiome studies in traditional populations. And what are these barriers? These barriers exist in every step from sample collection to publication. So there's very limited funding, or in many cases, complete lack of funding to look at microbiome of traditional population, in-country funding. Most of the funding in country or from international agencies focus on infectious diseases, and there's very limited understanding of microbiome and its effect on human health within the funding agency, regulatory agencies, and sometimes among the scientists in the low and middle income countries. It's also very difficult to collect samples because there is lack of health infrastructure. So there are not a lot of hospitals, and people live in remote geographic locations. So people don't usually come to hospitals until it's like an emergency situation, right? So collecting large number of samples is very, very difficult. And who will collect the samples? We need trained people to collect the samples, and it's very difficult to find trained field staff. And even if you go out there to train the field staff, there's a high turnover, and it's extremely difficult to sustain a trained workforce from personal experience. Whatever number of samples you collect, when you bring them to a university or a research institution to do lab work or genomics research, we have to face the scenario where there is very limited research infrastructure. We have very few trained scientists, and even if you train scientists, they also leave. Sometimes they leave to do PhD abroad. Sometimes they go and find another type of job. So it's very, very difficult to train, sustain a trained workforce. Bioinformatics analysis, the capacity to do that is virtually non-existent in many low and middle income countries. There's very limited computational infrastructure. There's not a lot of training provided at master's or undergraduate levels. So hard to find experts or trainees who are interested. And even if you train some people, again, they also leave for better jobs abroad. So all this culminates into limited number of samples that we can get, and one-dimensional data is what I call it. So basically people may do 16S or maybe some metagenomics, but just that, right? We cannot have, there's not a lot of opportunities to generate multi-omics data, and which equates to limited novelty. And limited novelty equates to less chances of publishing in high-profile journals, which results in limited funding for subsequent studies, and we go back to the top again, and this cycle repeats. So this happens at a very reasonable frequency when we start thinking about traditional and underrepresented populations. So what we have done in the past, since 2019, is we've tried to develop this systematic approach in scaling up functional studies in microbiome in underrepresented population. So we published a paper in 2018 in which we compared gut microbiome variation going from foraging populations on the left in this heat map to farming and industrialized populations. And we see massive shifts in gut microbiome. So farmers living in Nepal, their microbiomes are starting to resemble the industrialized populations, right? So there is this massive shift. So using this as a foundation, we have been successful in obtaining a grant from Bill and Melinda Gates Foundation, which allows us to go all the way to the functional level. So we're asking 3 questions. So we're conducting 2 studies looking at the functions of gut microbiome in underrepresented population. One is in Himalaya, one is in the Emirati population here in UAE. And the first question in the Himalayan study we're trying to ask is, how does the gut microbiome develop in infants across lifestyle? Because like I said earlier, the ecological progression is very well studied in Europeans, but we don't know how the progression happens in underrepresented traditional populations. So this is sort of like characterizing the gut microbiota. The next step is to look at association. So in Nepal, 33% kids actually end up stunted. It's because of undernourishment, other factors, right? So Gut microbiota are causally linked with stunting in other populations. So we want to ask, are there associations between gut microbiomes and stunting in Nepali children? And if there is, the third step is, what is the biological mechanism that leads to gut microbiome dysfunction in stunting? So to answer these questions, we're collecting a massive trial in Nepal, or it's not really trial, it's a observational study where we're collecting samples from pregnancy, first trimester in pregnancy. It's a longitudinal study to the end of first year of life in kids. So we're collecting maternal lifestyle and diet using survey questionnaire. We're collecting maternal gut or fecal samples for gut microbiome analysis, vaginal microbiome, and we're also collecting using survey questionnaires, if various factors that influence the child's life in the first year. So for example, child's birth mode, what this kid is eating, breast milk, formula, non-formula food, medicine, vaccination, and other sort of environmental factors. So the goal here is we want to use the metagenomic data or 16S data to understand how gut microbiome progresses. Maybe it's different in different lifestyle, and maybe this sort of the dysfunction will lead to some association between stunting and particular microbes. Once we figure out which microbes are associated with stunting or low birth weight, we can then extract those microbes, because we're collecting all of these samples using liquid nitrogen, so they're flash frozen. So we can isolate these strains and do functional characterizations in the lab. So here is our sampling scheme. We're collecting samples from 4 populations: foraging population, a farming population, a peri-urban population, and an urban population. On the top is actually the sampling scheme. So we're collecting samples from controls. Basically, these are women of reproductive age who are non-pregnant. So we're collecting samples in the 3 samples every 3 months, 1 sample every 3 months. And we're collecting samples from pregnant women at time point 1, time point 2, time point 3. Red dots here means fecal sample. Saliva, we're also collecting it. I'm not going to get into it. At birth, we're collecting vaginal swabs. And then for the first year of the child's life, we're collecting breast milk at 1 month interval. And then we're also collecting gut microbiome or fecal samples from infants. In the first 3 months, we're collecting samples at every 2-month interval. And after that, we're collecting samples at 1 month interval up to the 12th month. Despite COVID, we have completed sampling from the first trimester to the 6 months of the kid's life. We're collecting currently samples from 7th to 12th month, and we're hopeful that we'll get funding to continue this to the first 3 years of life. To sort of give you, oh my God, this is covered here, but to give you an idea of the scale of this work, we are collecting 50 mother-infant duos from each group. We have 4 groups, and there are 21 time points, which results to about 4,200 fecal samples that are flash frozen. We have also 200 vaginal samples, and we have about 2,400 breast milk samples. Collectively, this results to 6,800 biological specimens, and we're collecting samples in multiple replicate tubes, which results to, in our estimate, about 19,000 tubes of samples from these 4 underrepresented populations, which is a pretty massive number of samples to collect using flash frozen medium. So we are currently this is an ongoing study. It's not completed yet, but once this is completed, we're really hopeful that this will result in meaningful discoveries that will be useful in addressing stunting problem in Nepal. The other study that we're doing here is looking at the oral microbiome as a part of the UAE Healthy Future study, which was funded with a local investment funded by the UAE government. So for this study, we have collected DNA from, we collected spit samples from, mouthwash sample from 27 individuals from which we extracted the DNA. The optimization to extract DNA from mouthwash samples took us a little bit of time, but we've done it. We're currently doing 16S and metagenomics sequencing from these individuals. And we also have 300 clinical traits, right? So these are blood markers, anthropomorphic traits, other sort of disease-related markers from each of these 2,700 individuals. We're in the process of also developing metabolomics. I put a black box here because I have no idea how to work with the metabolomic data, but it is important. So we're sort of inching towards learning how to integrate metabolomics with the metagenomics data. So at the end of this study, the goal here is to identify link between oral microbiome and disease and understand the biological mechanism by which oral microbiome may influence the disease. So with that, the summary of our work in the last 3 years is that we know that human microbiome as an ecological process is very important, and we need to understand it in detail to sort of understand how microbiome and human health are connected. We need functional studies linking human microbiome to diseases in traditional populations because those types of studies are scarce. But there's urgent need to start these studies because these types of studies benefit the participating populations, right? So NCDs are rising rapidly in non-industrialized countries, and without these types of studies, there is a chance that that these countries, their health infrastructure may collapse if we cannot prevent NCDs from rising. Despite all these obstacles, I think these two studies show that it is possible to conduct large-scale functional microbiome studies that benefit these underrepresented populations. And this is especially possible if local stakeholders are involved, like if the government regulatory agencies are involved or funding agencies, local funding agencies are involved. With that, I want to thank, I have a lot of people to thank. I'm not going to go into each person, but there are a lot of people who helped me do the field work, are currently helping me. There are people involved in laboratory work. I definitely want to thank Biswas Chepang, the autocorrect messed it up. His name is Biswas Chepang, and the female community health volunteers in Nepal who have been massively helpful in recruiting participants. A lot of collaborators here, but I definitely want to acknowledge Justin Sonnenberg at Stanford, with whom we're running these some of these studies together. Funding sources, I cannot thank the Gates Foundation enough. And we're very, very, very thankful to the participants for welcoming us to the communities, as well as the Nepal Health Research Council, who gave us permits to conduct these studies, Stanford and NYU AD IRBs as well. So with that, I want to thank you for your attention. My lab is called Genetic Heritage Group. You can reach me by email or through Twitter. If you have any questions, we're open to collaborations. And if there's any recently graduated PhD students and looking for a postdoc, we're also looking for a few postdocs. So with that, I'll take questions. Thank you.
Thanks so much, Dr. Jha. That was a great talk. We have a little bit time for question, but probably don't want to take up too much of your time either to allow you your well-deserved rest. There was one question I saw in the chat that was asked by Ambery, who was asking what is being referred to when we say traditional microbiome studies versus European microbiome studies. Are we just referring to non-European populations?
Yeah, so the difference there is when we say, well, so sort of like European is synonymously used to indicate like industrialized population, right? Because most of Europe is pretty industrialized. So when we say traditional population, we're talking about populations that live in non-urban areas.
Awesome, thanks so much. Yeah, and yeah, I was also interested, about your experiences in terms of community engagement, like what did you think, for instance, like was particularly important there, but also like what additional information you might have gotten from engaging with community representatives and building up those collaborations? And yeah, that would be my question.
Sure, yes. So community engagement is super important, but how you define community engagement may differ, right? So in our study, those ongoing studies in Nepal, what we've done is we've hired people like our sample collectors are people from within the community. We have gone through extensive length to actually identify, nurses from that community, and we've hired them so that people feel comfortable, participating in our studies, right? And that not only from our perspective, but when you hire people from the community, they are willing to go an additional mile to help their own community. So that is super important, right? So in the past, what we did was we sat down with people, we talked to the community members, and we collected samples, but I did the sampling myself. So there was community involvement, but they were not involved, they were not actively engaged, right? Right now, we're hiring people year round to go and collect samples for us. We've trained them, and they're from that community, so they are stably there, and they're actually making massive differences that I had never thought about. So and I know we're short on time, but I think this is important, so I'll discuss this. So we were thinking about malnutrition, gut microbiome, identifying causal bacteria and all that, right? But one of the things that we did not realize was we can give back to the community in real time. So one of the things that we can give back to the community is Nepal government actually has a program. It's a government-related program. So all mothers who do 3 prenatal checkups and do an institutionalized delivery, so not delivering the baby at home, but in a hospital or a health care facility. If a woman completes all those 4 milestones, she actually gets like a few $1,000s from the government, right? And this money is not that important, but what is more important is it actually influences the child's health and the maternal mortality, both, right? Maternal and child improves those 2 things. So these women who we have hired, all of our sample field workers are women. They actually are checking and making sure that these women, our participants, are going to are sort of like fulfilling these milestones. And as a result, we have like about 80% of our participants have actually fulfilled this milestone. And overall average is like around 30% or 40%, something like that, right? So that is not something that we had thought of before we started this. This just came organically from our team who's working in Nepal, and just like, it's a massive contribution to the participating community, which I'm very, very proud of.
Yeah, thanks so much. Yeah, maybe we have time for one more question, and then you'll be off to be able to sleep finally. But there was a question by Noura El Habal, who was asking about the infant microbiome study. So she was asking if you have data on maternal diet or medication, and then also about the whether is also measured during lactation. I might have missed this, but during which times do they collect milk samples, and do they take into account infant feeding?
Yes, so we do have data on the mother. So we actually have 4 different survey questionnaire. Each survey questionnaire has 150 to 300 questions. It's massive, I know. Our program officers, they complain about it, but the point of using the survey questionnaire is there is no data from these populations, right? Like nothing. Like we don't know what they do during pregnancy. Is there a special diet that they use? What happens in the child's first year of life, how much interaction they get with like animals, with other. So basically we have 1 survey data to look at the pregnant women. And then we have another survey looking at the child in the first 3 months. And then a third survey data from 3 to 6 months. And then a 4th survey questionnaire to capture lifestyle related covariates from 6 to 12 months, right? So in using this survey questionnaire, we can get a good sense of what the mothers and infants are eating, whether they were vaccinated or not, whether they were medicated or not, and so on. So what time the breast milk sample is being collected? All of the samples are being collected in the morning, because people live in very remote areas, and we don't have the luxury to just keep visiting the same person multiple times during the day. So our field officers, they go to the participant's home before 6 a.m. So the participant produces the fecal samples, and then, she just hands the fecal samples to our field officers. The fecal samples are dropped in a liquid nitrogen tank. We have a small portable liquid nitrogen tank that that people carry door to door. And then the breast milk is also collected at that time. So we don't have an estimate of whether, it was a recently fed milk or not. But yeah, that's one of the limitations.
Yeah, perfect. Yeah, and with that, let me thank you again for taking the time to speak to us today. It was like a huge privilege and an amazing talk. And a huge virtual applause to Dr. Jha from our participants here. And thanks for joining us today.